Hello dear students. Today we are going to learn about the red cell shapes. First is spherocytes. Spherocytes are dense, small red blood cells with reduced mean corpuscular volume and increase in the mean corpuscular hemoglobin concentration. They appear as dense red blood cells with loss of central pallor and they can be seen in the and these cells can be seen in the hereditary spherocytosis, hemolytic anemias, and clostridial sepsis. Next is the teardrop cells or dacrocytes. Teardrop cells appears as a drop of tear and they can be seen in the myelofibrosis, iron deficiency anemia, thalassemia major, and myelodysplastic syndromes. Now come to the next are the cystocytes. Cystocytes means the fragmented red blood cells which can be seen due to the mechanical effects on the red blood cells such as seen in the prosthetic heart valves, severe sepsis, disseminated intravascular coagulation, and MAHA, that is the microangiopathic hemolytic anemias, which can be seen in the thrombotic thrombocytopenic purpura, hemolytic uremic syndrome, HELP, that is uh, that is the complication of eclampsia. Now come to the echinocytes. Echinocytes are also known as birth cells. These cells have the small evenly distributed spicules over the red blood cells. The spicules are small and evenly distributed. These cells can be seen in the uremia or it can be artifact due to the prolonged drying. Now come to the acanthocytes. Acanthocytes are also known as spur cells. Spur cells have large spicules and these spicules are not evenly distributed, and these acanthocytes can be seen in the liver disease, neuroacanthocytosis, a beta lipoproteinemia, and severe malnutrition. Next is the elliptocytes. Elliptocytes are elliptical red blood cells which can be seen in the hereditary elliptocytosis, which is a defect due to the spectrin, protein 4.1, and band 3, and sometimes glycophorin C also. Other causes of elliptocytes are iron deficiency anemia, myelodysplastic syndrome, and thalassemia. Another red blood cell shape is stomatocytes. Stoma means mouth cells with a mouth-like opening in the red blood cell. As we can see in these cells, they have central area of pallor which appears as a mouth. So it is seen in the hereditary stomatocytosis, which is of two types, hydrocytosis or xerocytosis. Cirrhosis, alcoholism, and Rh null disease. Next is the codocyte or target cells. Target cells form due to the increase in the red cell membrane amount as compared to the volume. So the central area appears as a accumulation of hemoglobin in the center and there is loss of central pallor, and this is seen in the thalassemia, hemoglobinopathies like HbC, HbE diseases, post-splenectomy, liver disease, and it can also be an artifact. Now, another thing which we can see in the red cells is the agglutination. Agglutination is the clumping of the red blood cells, which is seen commonly in the cold agglutinin disease. Cold agglutinin disease is seen in the lymphoid malignancies like chronic lymphocytic leukemia, infections like mycoplasma infection, EBV infection, which causes infectious mononucleosis, and cytomegalovirus infection. Another cause is the cryoglobulinemia. Cryo means cold globulins, which precipitated during the cold weather and causes the agglutination of the red blood cell. Artifactual causes can also be seen in some cases. Rouleau formation is seen in the multiple myeloma. It is the stacking of the red blood cell over each other, and it is commonly seen in the paraproteinemia and multiple myeloma. Paraproteinemia means increase in the clonal immunoglobulin, which is commonly seen in myeloma and paraproteinemia. Other cause is the chronic inflammation, which causes increased immunoglobulins, but these are polyclonal. So, in the hypergamma globulinemia state, these Rouleau formation can be seen. Another is the sickle cell. Sickling of red blood cell is seen in the HBS or sickle cell disease. In the sickle cell formation, the dense hemoglobin S crystal precipitate and changes the shape of the red blood cell to form a elongated sickle-like structure which does not has central pallor, no central pallor. Remember, you can confuse with the pencil cells, which is actually seen in the iron deficiency anemia. Pencil cell and sickle cell can look similar, but pencil cell has a central pallor, and pencil cell seen in the iron deficiency anemia. However, sickle cell does not has any central pallor. So this was all for today. Thank you.